Chromosomal aberrations. UGC Net Anthropology. Part 2. Welcome to AnthroNet Academy, a study aligned with the UGC Net Anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Big surprise at 500 subs. Once AnthroNet Academy hits 500 subscribers, we're dropping something truly special just for you. A full video revealing 100 powerful, underrated, and must-read books list in anthropology, complete with author names and insights important for UGC Net exam. Structural aberrations. The structural aberrations in chromosomes fall into 4 major categories. Each of these changes can be triggered by one or more breaks on the chromosome, followed by rejoining of the broken ends to generate new chromosomal arrangements. For types of structural mutations, deletions and duplications involve a change in the amount of DNA, and the other two bring about a change in the orientation, inversion, or location, translocation, of a chromosome segment, with no change in the amount of DNA. Deletions. Deletions are a type of chromosome mutation characterized by loss of one or more segments of the chromosome. A deletion can be induced by various agents such as ionizing radiations, transposable elements, or by imprecise recombination, generates one chromosome with a deletion and another with duplication. Depending on the part of chromosome deleted, a deletion may be terminal or interstitial, intercalary. The terminal deletion requires a single break, while the interstitial, intercalary, deletion produces 3 fragments by introducing 2 cuts and then joining 2 of the terminal fragments' ends. Karyotyping can easily detect a large deletion because the chromosome is shortened. In addition, during prophase I of meiosis, individuals heterozygous for deletions align by looping out the undelete segment, deletion or compensation loop, of the normal chromosome. The consequences of a deletion depend on which genes are located in the deleted segment and the size of the deletion. If the deleted segment includes the centromere, then the chromosome will not be segregated during cell division and will eventually be lost. In the amount of gene products, haploinsufficiency, or pseudodominance. Example: in humans, a deletion on the short arm of chromosome 5 is responsible for Cri-du-Chat syndrome. 2. Duplications. Duplications involve duplication/doubling of a part of the chromosome. The size and location of duplicated parts vary. Depending on the location, they are classified as tandem reverse or displaced duplications. They are generally caused due to imprecise recombination. Types of duplication. Tandem duplications. In tandem duplications, the duplicated part and the original sequence are adjacent to each other. Displaced duplication. If the duplicated segment is not in tandem, but away from the original sequence in the same or different chromosome, it is called displaced duplication. Reverse duplication. In some duplication, the orientation of the duplicated sequence is inverted as compared to the original segment, which results in reverse duplication. Example: an example of gene duplication involving a small segment on the X chromosome of Drosophila melanogaster is the bar mutation. The mutant flies have fewer facets in the compound I, giving it a bar shape. It is inherited as an incompletely dominant condition. 3. Translocation. A translocation entails the movement of a chromosomal segment to a new location in the genome, but the amount of genetic material remains unchanged. It may be either reciprocal or non-reciprocal. Reciprocal versus non-reciprocal translocation. Feature: reciprocal translocation. Non-reciprocal translocation. Number of chromosomes: 2. 2, but only one gives material. Exchange type: mutual, balanced. One way, unbalanced. Genetic material loss/gain: usually none, if balanced. Often results in imbalance. Effects on carrier: often silent. Often problematic. Example: T1, 9, Q34, Q13. 5Q15Q, no return from 15. 4. Robertsonian translocation. Robertsonian translocation is a type of reciprocal translocation first described by William Robertson in grasshoppers. It is one of the most common chromosome rearrangements in humans involving acrocentric chromosomes 13, 14, 15, 21, and 22. Examples: in human conditions due to reciprocal translocation are familial Down syndrome, Burkitt lymphoma, and chronic myelogenous leukemia. 5. Inversion. The last type of chromosome rearrangement is an inversion in which a chromosome segment is inverted. For an inversion to take place, the chromosome must break at 2 places and then rotate 180 degrees prior to rejoining. The inverted segment may be of variable length, which may or may not include the centromere. Types of inversion: if the centromere is not a para equals next to, and when the centromere is also included, it is classified as paracentric inversion, peri equals around. 6. Isochromosomes. Isochromosomes are unbalanced, structurally abnormal metacentric chromosome formed by the duplication of one arm of a normal chromosome with deletion of the other arm. Both arms of the metacentric chromosome are thus genetically identical, mirror image. Ring chromosomes. A ring chromosome is an aberrant chromosome whose ends have fused together to form a ring. It was discovered by Lillian von Morgan, 1926, in Drosophila. They are formed by breaks at the ends of both chromosome arms and rejoining of the broken ends, or by fusion of dysfunctional telomeres from the same chromosome. Such a chromosome may be formed from any chromosome and is generally accompanied by loss of genetic material, variable size, and mitotic instability. Causes of chromosomal aberrations: errors during meiosis or mitosis. Non-disjunction or faulty recombination can lead to structural or numerical changes. Exposure to radiation. Ionizing radiation, like X-rays or gamma rays, can break DNA strands. Chemical mutagens. Substances like benzene, pesticides, or chemotherapy drugs can alter chromosome structure. Viral infections. Some viruses can integrate into host DNA, causing breaks or rearrangements. Inherited genetic defects. Mutations in genes involved in DNA repair or chromosome segregation can lead to instability. Environmental toxins. Heavy metals, pollutants, and industrial chemicals may damage chromosomes. Advanced maternal or paternal age. Increases the risk of non-disjunction during gamete formation. Spontaneous mutations. Random errors during DNA replication or cell division. Thank you for watching. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.